Introducing
Serological test and its purpose. Serological test is antibodies in your blood can be detected through blood tests. They can involve a number of laboratory techniques to diagnose various disease conditions. Different types of serologic tests need to be used. The purpose it is helpful to know about the patient's immune systems. The substances that provoke a response from the immune systems are called as antigens. They can easily enter our body, such as through our broken skin, as they are very tiny. Furthermore, the immune system will produce antibodies, and these antibodies will attach and deactivate antigens. When the doctor tests the patient's blood, they can identify the types of antibodies, antigens, and infection that they have. Sometimes, outside invaders and unnecessary antibodies have been produced, and it known as an autoimmune disorder. So, this serology testing can detect these antibodies and to diagnose an autoimmune disorder. Of serological test. He is Jules Bordet. He is Belgian physician, bacteriologist, and immunologist. He discovered the factors in blood serum that destroy bacteria, and this work was vital to diagnose and destroy many dangerous contagious diseases. Bordet's research on the destruction of red corpuscles and bacteria in blood serum contributed to the foundation of serology. In 1895, he found that two components of blood serum are responsible for the rupture of bacterial cell walls. In 1898, he discovered that red blood cells from one animal species that are injected into another species are destroyed through hemolysis process analogous to bacteriolosis. Then he continued his research with his brother-in-law, which led to the development of the complementization test. This technique is used to detect the presence of infectious agents in the blood and those that cause syphilis. After discovering the bacterium, Bordet became professor of bacteriology at Free University of Brussels.
Okay, so right now I am gonna talk about procedures of serological tests. Firstly, there are many serological tests out there, but I am gonna talk about two commonly used serological tests. The first serological test is rapid serology test. Firstly, we will collect a blood sample from a patient, and then we will add the blood sample to the sample well. After that, we will place 2 to 3 drops of buffer in the sample well and read the results after 50 minutes. And lastly, the results was interpreted and recorded. The second serological test is enzyme-linked immunosorbent assay, also known as ELISA. The first step is the antigen is adsorbed onto well in ELISA plate in coating buffer, and then the buffer was removed and the plate was washed. The third step is a buffer containing unrelated protein is used to block free sites in the wells. And again, the buffer was removed and the plate was washed. The fifth step is enzyme conjugated detection antibody binds antigen was placed to detect the antibody. And the buffer was removed and the plate was washed. The seventh step is the substrate was catalyzed by enzyme to generate colored readout. And lastly, the result is then observed and recorded.
Now we're going to talk about the application of serological tests. Firstly, it is to study the development of autoimmune diseases. The serological test is useful in aiding the detection of antibodies produced by past or current infections. After that, serological test is useful to study the effectiveness of vaccines and the subsequent level of antibodies production. The serological test is helpful in evaluating the effectiveness of COVID-19 vaccines. After being vaccinated, a person's immune system will develop and produce antibodies against the microorganisms that is contained in the vaccine. After being introduced with the virus parts, the immune system will induce antibodies and the level of antibodies will increase after the next few days. The following graph will show you the level of antibodies against COVID-19 after the vaccination. The serological test could help physicians in evaluating the current or past infections of COVID-19. This test is also useful in detecting antibodies to check for current or past bacterial or parasitic infections. Syphilis is one of the sexually transmitted disease STDs caused by the bacteria Treponema pallidum. To have a positive reaction in serological tests in syphilis serology test could indicate a person has been infected with Treponema pallidum and has developed antibodies that could be detected in the patient's serum. Next, chlamydia trachomatis that could cause chlamydia, which is a common STD. If a patient has developed antibodies against chlamydia trachomatis, this could indicate that the patient has past or current chlamydia infections. The same could be applied in detecting antibodies produced in Toxoplasma gondii, which is a type of parasite. The serological test could also help in studying various types of autoimmune diseases. For example, we could study the development of rheumatoid arthritis, which is a chronic joint disease caused by autoantibodies that attack a person's own tissues instead of attacking foreign pathogens.
Limitations of serological tests. The limitations are: first, it will increase risk of cross-reactivity reaction. Second, serological test is only focusing on patient's immune system instead of the disease itself. Third, poor sensitivity or inadequate antibody levels in patient sample. Fourth, biological problem causing difficult in deciding tests, hard to interpret result, and troublesome to standardize test system. Lastly, it can also cause false negative results.
Throughout this video, we can conclude that serological tests are used to determine whether a person had a past exposure to a pathogen. The tests look for antibodies which can bind to a pathogen that will form as part of the immune response. Serology tests use blood samples which collected from either a blood draw from a person's arm or finger stick. Other than that, different types of serologic tests need for diagnosing various types of disease such as syphilis, COVID-19, rubella, allergy, and HIV. Apart from that, this test has been used in several applications like Ashla has explained. Nevertheless, even some serology tests is easy to run, there are still some limitations which Sarah Christina has explained.
That's all from us. Thank you.